Category number 2, agranulocytes. These are cells with no visible granules in their cytoplasm under a light microscope, and they are mainly involved in adaptive immunity and regulation. Examples include number 1, lymphocytes with dark purple, round, slightly indented nucleus, with clear sky blue cytoplasm in small lymphocytes. Lymphocytes are divided into B cells, T cells with very little cytoplasm and dense nucleus, and the natural killer cells, which are the large or activated lymphocytes with more cytoplasm and mostly condensed nucleus. The B cells or the B lymphocytes develop in the bone marrow and responsible for humoral immunity, targeting extracellular pathogens. The B cells differentiate into plasma cells, which produce antibodies, and memory cells that provide quick response during reinfection. Number 2, the T lymphocytes that mature in the thymus and responsible for cell-mediated immunity, targeting intracellular pathogens. The T cells are further subdivided into number 1, helper T cells or the CD4 cells, which activate the B cells, cytotoxic T cells, and macrophages. Number 2, cytotoxic T cells, the CD8 cells, which kill virus-infected cells and cancerous cells. Number 3, regulatory T cells, which suppress autoimmunity and control immune response. Number 3, the natural killer cells, which are part of the innate immune system that kill virus-infected cells and tumor cells. So the lymphocytes contain number 1, receptors, which recognize antigens or abnormal cells triggering immune response. For example, the B cell receptors found on the B cells and recognize specific antigens, usually in their native form. So once activated, the B cells proliferate. Number 2, differentiate into plasma cells that secrete antibodies matching the B cell receptors. Number 2, T cell receptor found on the T cells. So recognize antigens presented on major histocompatibility complex molecules. So the MHC1 for cytotoxic T cells and MHC2 for helper T cells. Therefore, their activation leads to cytokine release or killing of infected cells. Number 3, natural killer cell receptors found on natural killer cells. So these detect the absence of self markers and common in virus-infected or tumor cells. So these receptors balance between activation and inhibitory signals, determining natural killer cell action. The second component of lymphocytes are mitochondria and Golgi bodies. These organelles help, for example, the mitochondria provide energy for cell survival, proliferation, and function. The mitochondria are active during lymphocyte activation, cytokine production, and cell division. The Golgi bodies packages and processes cytokines and effector molecules for secretion. They are especially important in plasma cells making antibodies and T cells producing interleukins or cytotoxic granules. Component number 3 are cytotoxic granules, majorly found in natural killer cells and cytotoxic T cells. These granules contain molecules that kill target cells, especially infected and cancerous cells. Molecules include granzymes, a family of serine proteases that enter the target cell through pores made by perforins. So they trigger apoptosis, which is programmed cell death by activating caspases and destroying DNA and other vital proteins. Cytotoxic granules also contain perforin, which is a protein that creates pores in the target cell membrane, allowing granzymes to enter the cell, leading to programmed cell death. Second agranulocyte is the monocytes and the macrophages. These appear light purple with kidney or horse-shaped nucleus, with a dull gray-blue cytoplasm with possible fine granules or vacuoles. So monocytes circulate in blood and migrate into tissues and become macrophages or dendritic cells. So monocytes contain lysosomes, which are small bound sacs inside the cell filled with acidic enzymes like lysozyme, acid phosphatase, proteases, and lipases. So after phagocytosis or engulfing of the microbe, the lysosomes fuses with the phagosome, forming a phagolysosomes, and break down bacteria, viruses, dead cells, and debris. They destroy pathogens using reactive oxygen species and enzymes. Number two, surface receptors, which are protein on the surface or inside the monocyte or macrophages that detect specific patterns found on pathogens, known as pathogen-associated molecular pattern. These trigger activation of immune cells, induce inflammatory responses, and stimulate cytokine production and phagocytosis. Number three, major histocompatibility complex 2 molecules, which are proteins found on the surface of antigen-presenting cells, like the macrophages, dendritic cells, and the B cells. After digesting a pathogen, macrophages display small pieces called antigens on MHC2 molecules. These are presented to helper T cells, the CD4 cells, hence activating adaptive immunity, prompting the T cells to release cytokines or assist B cells in making antibodies. Number four, cytokines. For example, interleukin-1 and tumor necrosis factor alpha. These are small proteins released by macrophages to signal other immune cells. For example, interleukin-1 induces fever by acting on the hypothalamus. Number two, promotes inflammation. And number three, activates T cells and endothelial cells to allow more white blood cells migrate into tissues. Tumor necrosis factor helps recruit more immune cells to infection sites.